Thank you so much, Eyecelerator, for putting these together. And it's my first time presenting here, so I'm really grateful. You know, vision is 80% of our perception of the world is vision. So what we do here is truly monumental and tremendous for everyone around the world. So thank you for all of you and all your work you do. So this is actually my grandmother. My grandmother had macular degeneration. This is a serious disease. I had to drive her to the doctor once a month. At the time, Lucentis was approved in Canada, and I can tell you she was totally healthy. And like her, many patients after year one gave up on that frequency of injection, which is quite intrusive. And so we know that most retina specialists are looking for a better solution, less injection, to have better compliance. Our vision is to have a very simple, elegant solution, and we've developed a set of amino acid sequences that you can add to any biologic. Imagine that, at a genetic level, that can grow out of a CHO cell line, and it could come out, and it would be just like, for example, Eylea in terms of viscosity, using a 30 kg needle, volume, everything else. And potentially you could use this set of amino acid sequences for any biologic, so another complement inhibitor or bispecific, and so on and so forth. We have shown a proof of concept, and I'm delighted to share with you our non-human primate study results that we just completed. Our lead asset is XPK640. We're preparing for IND at this point. We've developed a platform. We have an experienced team. Primarily, our team comes from iBio, several of them from Allergan, as well as Iveric. And we are looking for partners. We're already in discussion with several partners that we're helping with their other biologics. We all know this. This is a graph that's been shown by our peers. Frequency of injection. The moment you have less injection, you can actually have a better treatment, and ophthalmologists definitely prefer that. Our target product profile looks like something like this, and I can tell you that we've moved our target product profile over the last few months. We were aiming for something like 6 to 9 months frequency of injection. We've now moved to 9 to 12 months. We're hoping to have the same efficacy and safety of Eylea, but actually be able to inject it once a year. That's the vision, and potentially other biologic. So, how do we do this? We're using eye's own polymers, in this case hyaluronic acid, which is very abundant in the eye. So we've actually come up with a set of amino acid sequences that literally at a genetic level we add to another biologic, in this case aflibercept. We grow this out of a CHO cell line, and these amino acid sequences actually have affinity to hyaluronic acid. Now, we've gone through about 4 or 5 years of screening collagen type 2, albumin, and many other sequences around hyaluronic acid, and tested many of them in rabbit studies. And XPK640's specific set was the best one because we had to come up with one that's not too sticky and it's not too loose. It actually has to be a Goldilocks to create this perfect equilibrium so it doesn't get stuck. This is what the molecule looks like in this case. So this is the bottom part, which is the hyaluronic binding. As I said, it's a recombinant expression performed in mammalian cell line. It's done at a genetic level. It's compatible with other biologics, again, either bispecifics or trispecifics. We've already done 2 additional biologics, but I'll share with you those with you as well. And the molecule doesn't become significantly larger. Again, we do not need extremely complicated polymer chemistry. We do not need light, heat, anything like that. In this case, aflibercept is about 100 kilodalton. Our molecule will be about 120 kilodalton. So we ran a pilot tox PK study. Actually, it was designed by one of our pharma peers here, which was a fantastic study. And I'm just showing you the PK, and I'm going to show you all the results from the tox as well. This is what the PK looks like. We were able to expose 100 to 1,000-fold more exposure in this case, and actually be able to extend the PK by about 10x. Half-life 36 days versus 2 to 3 days in aqueous humor. Possible once-a-year injection. This is the top line overall, what the study results were. We were able to actually show tox findings were very similar to other approved products. No retinol toxicity, no systemic exposure, no precipitation or aggregation were observed, and VEGF binding affinity was maintained long-term as assessed in aqueous humor. Significant prolonged efficacy was shown in earlier VEGF challenge studies that we've shown in rabbit studies last year. Again, this is a modeling we've done, potentially once a year. And these are the other products in our pipeline, an anti-C5 as well as an anti-ILR6. We are now moving towards a clinic, hoping to be in the clinic next year. We've met with the FDA. They've approved our plan both for IND as well as preparation to go into pivotal trials after our phase 1/2A studies. I'm out of time. Thank you so much.